Welcome, everyone. Today, we're diving deep into a class of antimicrobials that truly changed the world, the penicillins. They're one of the absolute cornerstones of modern medicine. But how do they really work? And maybe more importantly, in this era of increasing resistance, how can we be smart about using them? That's what we're here to unpack.
All right, so here's how we're going to tackle this. First, we'll cover the basics in our Penicillin Primer. Then we'll jump into the real-world stuff, indications and dosages. After that, we'll look at the risks and contraindications, which is crucial. We'll also spend some time on sensitive populations, like in pregnancy and neonatal care. Then, we have to talk about the elephant in the room, resistance and stewardship. And I'll wrap it all up with a quick knowledge check to make sure the key points have landed. Sound good? Let's jump in.
Okay, let's dive right into our first section, the Penicillin Primer. This is all about laying the groundwork.
So, to start at the very beginning, what exactly is a penicillin? Well, at its core, it's a bactericidal beta-lactam antibiotic. Now, bactericidal is key. It means it actively kills the bacteria. It doesn't just stop them from growing. And it does this by going after a very specific process, the synthesis of peptidoglycan. This is the stuff bacteria absolutely need to build and maintain their cell walls.
And this is a fantastic way to think about it. It's like trying to build a wall without any bricks. You can imagine what happens, right? The whole structure is compromised. It can't hold its shape, and eventually the cell wall just falls apart. The cell ruptures, and that's the end of the bacterium. It's an elegantly simple yet devastatingly effective mechanism of action.
Now, this next point, this is something I really want you to take away from our discussion today. A reported penicillin allergy is not a diagnosis. Think of it as a hypothesis that needs to be tested. Why? Because the data is pretty staggering. The vast majority of patients who think they're allergic to penicillin actually aren't. So before automatically switching to a different, perhaps broader spectrum antibiotic, it is absolutely essential to question and, where possible, test that allergy claim.
Okay, moving on to our second section. Now that we've got the fundamentals down, let's get practical and talk about clinical application, the indications and dosages.
And just look at this table. It really highlights the incredible versatility of the penicillin family. You've got everything from Benzylpenicillin, given as a continuous IV for really serious infections like necrotizing fasciitis, all the way to Amoxicillin, a workhorse for childhood pneumonia. Then there's Flucloxacillin for those common skin infections, and even long-acting intramuscular forms like benzylpenicillin for something like syphilis. It's this range that keeps them so central to our practice.
Let's put this into a real-world context. Picture a patient presenting with necrotizing fasciitis, a truly frightening, fast-moving infection. In that situation, high-dose, continuous intravenous benzylpenicillin isn't just an option. It's an absolute cornerstone of the treatment plan, working hand-in-hand with urgent surgical debridement to try and get ahead of the infection. All right, let's shift gears now to Section 3. No medication is perfect, so we have to talk about the risks and contraindications. This is all about navigating potential harm. So when we talk about side effects, we have to look beyond the typical allergy concerns. For instance, with amoxicillin, you might see a specific kind of rash, this acutely appearing generalized exanthema with pustules. Or with co-amoxiclav, while it's rare, you have to be aware of the potential for idiosyncratic acute liver failure. And of course, urticaria or hives is always on the radar. The key is knowing what to look for and what to tell patients to report. And when it comes to true contraindications, the details really, really matter. It's not enough to just say allergy. We have to distinguish between the types. If you have a patient with a severe, immediate type allergy like anaphylaxis, the rule is to avoid all penicillins and other beta-lactams that have similar side chains. But, and this is a critical distinction, if the reaction was a severe delayed type 1, like Stevens-Johnson syndrome, the recommendation is much broader. You have to avoid all beta-lactam antibiotics. Getting this right is paramount for patient safety.
Next up, Section 4. Here, our focus narrows to managing some of our most sensitive patient populations, specifically pregnancy and neonatal care. Let's talk about treating syphilis during pregnancy. This is a situation where penicillin isn't just the best choice, it's really the only choice.
It's the only agent we have that reliably treats both the mother and the developing fetus. That's why this multi-dose regimen of benzathine benzylpenicillin is non-negotiable. And it's also why if a patient reports an allergy, we have to go the extra mile with formal testing and, if needed, desensitization. There is simply no room for error here. And that importance carries right over into neonatal care. For a newborn with suspected early-onset sepsis, for example, our go-to empiric therapy includes amoxicillin. Why? Because we need to cover for key pathogens like listeria. It provides an absolutely critical shield for these incredibly vulnerable little patients.
Okay, which brings us to section 5, and this is a big one. We're going to tackle the modern challenge of resistance and the vital practice of antimicrobial stewardship.
Just look at this number. 80%. That's a huge figure. What does it represent? It's the approximate percentage of Staphylococcus aureus strains out there that are now resistant to penicillin. They produce an enzyme called penicillinase that just chews up the antibiotic before it can do its job.
And seeing it visualized like this really drives the point home, doesn't it? That massive red slice shows just how big the problem of resistance is for this one particular bug. This is exactly why, if you suspect a staph infection today, you can't just reach for basic penicillin anymore. You have to use a penicillinase-stable antibiotic, something like Flucloxacillin, to have a fighting chance. So, what's our strategy against this rising tide of resistance? It comes down to smart antimicrobial stewardship. And a core principle of that is de-escalation. The idea is simple. When a patient is critically ill, you start with broad-spectrum therapy to cover all your bases. But, and this is the crucial part, you get your cultures. As soon as you identify the specific pathogen and you know its sensitivities, you pull back. You de-escalate to the most narrow, targeted therapy possible. It's like switching from a shotgun to a sniper rifle. It's precise, effective, and helps preserve our broader antibiotics for when we truly need them.
All right, we're almost at the end. For our final section, let's do a quick clinical knowledge check to help solidify some of these key concepts.
Okay, first question for you. Which of the following is explicitly mentioned as a contraindication for penicillin use? Is it A, renal insufficiency, B, liver failure, C, hypersensitivity to penicillins, or D, using anticoagulants at the same time? And the correct answer is, of course, C, hypersensitivity to penicillins. While you might need to adjust your dose for something like renal insufficiency, a true documented hypersensitivity is the absolute contraindication that applies across the entire class.
And for our last question, let's think about resistance. What's a primary mechanism that bacteria use to become resistant to penicillin class antibiotics? Is it A, increased drug excretion, B, production of inactivating enzymes, C, altering membrane permeability, or D, decreased metabolism?
The answer here is B, the production of inactivating enzymes. This is a major strategy for bacteria. They produce these enzymes, most famously the beta-lactamases, which essentially act like molecular scissors, cutting the antibiotic apart and rendering it useless before it can even get to the cell wall.
And I want to leave you with this final, crucial thought. In an era where we're seeing more and more resistance, we have to remember that the most potent feature of any antibiotic isn't just its spectrum of activity. It's the clinician's judgment. It's our ability to choose wisely, to de-escalate appropriately, and to question everything. That's what will truly preserve the power of these incredible drugs for the future.